Hello! In today's lesson we're going to have a look at clinical testing and later clinical trials. We're starting with the historical perspective using a story of William Withering and his discovery. We're going to become a bit more modern and explain the main phases of clinical testing, and then we need to compare the two and work out which is better overall and why. So, let's start with history here. So plants have long been used before we had modern medicine in order to make various cures or treatments for different diseases. And even in the modern day, about 75% of them as an estimate of medicines are derived from different plants and plant extracts. Now, plants produce chemicals as part of their defensive system against pests, even against other plants nearby sometimes, and some of them are harmful to humans, and some of them are useful in the right dosages but need to be calibrated carefully. So we have to test different plant extracts for toxicity and how effective it will be. So let's start with the story of William Withering and his experiment. There is an extract from a textbook that my students have access to. If you're watching from elsewhere, this is the Salters-Nuffield AS/A-Level Biology Book 1, and it would be useful for you to go to page 191. I have a grid here with lots and lots of different questions based on the story. So what I'd encourage you to do is read the story first on page 191, then look at the questions and try and match up your facts. Pause the video and have a nice read. Okay, let's have a look at some answers. So starting off, the plant that William Withering investigated is foxgloves. Symptoms that occur when you eat the leaves of foxgloves are from a range. You could have dizziness, vomiting, hallucinations, or even heart failure. But the plant extract can be used to treat dropsy or edema. Now, the first two patients that William Withering treated with foxgloves were a brewer who fully recovered and an old woman who nearly died in the process of him working it out. He called his mixture Digitalis soup, and the name of the active ingredient, when we identified it nowadays, we call it digitalin. Now, edema as a condition can be caused by heart or kidney problems, and it's an issue where fluid builds up in your tissues, so there's a bit of swelling. In his hospital in Birmingham, there were 163 patients. They used lots of repeats in order to ensure reliability, and ideally an even larger sample size should be used. He found the correct dosage of digitalin by slowly increasing the dosage of this plant extract until the patient had diarrhea or vomiting, showing they were having side effects from taking it, and then they reduced the dose slightly so that they weren't experiencing the side effects anymore, and this is how he came up with his effective dose of digitalin for his patients. But ethically, this is a bit shady because he didn't know what the side effects could be. Some patients died during the procedure as a result of the side effects maybe being too severe if the dosage was initially brought up too high, and as the patients couldn't give informed consent because they didn't know the side effects either, it's rather we disapprove in the modern times of this. So we can improve drug testing today to make it safer and more effective. And this all starts when you give a compound and you identify it as medically useful, and then you would start your series of trials. There are 5 different stages that we'd look through, from preclinical testing, and then we have clinical trials with 3 different phases. We have an after-licensing trial, and finally we can talk about the different reasons why we do this. Again, if you are in my class, you should have access to page 192 of the SNAB A-Level Biology textbook. If not, pause and have a go at filling this in. We want to summarize in a particular table on page 192 to pull out the key points. So, let's have a look through. Now, preclinical testing is using animals and isolated cells and tissues, think petri dish cells, in order to find out if there is an effect. We're using more simple life forms here because we want to assess safety and effectiveness, and we need to work out how toxic this component could be. If the treatment is safe and it has a low toxicity, or if we know how toxic it is and can then try and work out what dosage to give, then we can move up to small groups of healthy people, so between 20 and 80 individuals. And this is useful because we can check how the medicine, how the compound is being absorbed, distributed, and metabolized and excreted from the body as predicted. So we're really just seeing how the body processes this component, even if you are healthy. We can look at the effect of adding different dosages to hang out for side effects. So if you expect a large dosage of something to have many side effects, this will help pick out what counts as a larger dosage. And then you can do an independent review following this to bring all your evidence together and make further decisions. And then you enter phase 2. And in phase 2, we're using small groups of patients again, but this time these are not healthy patients, these are people with a disease, and we're looking for between 100 and 300 individuals. So, carefully by administering the dosages, using all the information from before, we can look for the effectiveness in real patients. So how well is it treating those individuals? Do you see an effect? But also, do you see any negative side effects? So now you've got some information about how healthy people process it and how that molecule works in the body. We then have more information from patients with disease and how well it might work as a treatment, and now we can really start rolling it out. So we can recruit a larger group of patients, between 1,000 and 3,000, and this is to find out if there is a significant improvement in terms of statistics. We want to see a statistically significant improvement, and we're going to compare giving them this treatment to using a placebo, which is an inert substance or tablet or injection that you're providing. And you can compare the effects of the drug versus someone who thinks they're taking it, but in reality they are not, and you would observe the placebo effect. It's also double-blind and randomized, and if something is double-blind, it means both the scientist and the patient don't know what treatment they are giving that patient. So there will be less bias on the patient side and also the scientist side when they're observing and recording their data. They're randomized because the people who are receiving either the placebo or the medicine have not been picked or selected, therefore introducing bias by somebody making choices. Instead, they find a randomized measure in order to put people into these two groups. So now you've got lots and lots more data. You've really scaled up the operation and you've given it to your large group of patients. So after this drug has been licensed, then you'd keep on collecting data on effectiveness and safety, usually by asking people to report side effects if they observe any, or if they're in hospital, then side effects can be picked up that way too. So let's start putting some of our knowledge from this lesson together now. We have a practice exam question here where you have 3 marks, so that's 3 different things you need to say, and you need to describe, which is just saying what. Oh, there we go. Oh no. That's better. So describe and explain, say how or why, the procedures that must be followed before these drugs can be tested on patients. If you feel confident, hide your notes from before and just try and do this by memory. Otherwise, you're welcome to have a quick peek. So think, what happens before you give a drug to patients? What are those procedures that are followed? And explain why they're followed. 3 marks, off you go. Okay, so let's look through some answers. So we should have started with that very first level on our table where we're testing this medicine on non-human, so animals or tissue cultures, cells. And the reason why you're doing this to meet the explain criteria is that we're testing for toxicity, or testing for how damaging it is, or testing for side effects. Following this, because remember, patients is not one of the first 2 steps. Patients start getting involved after you've tested healthy volunteers. You could not say healthy patients, because again, we're looking for before that step. And this is to find out if there's any side effects. So score yourself out of 3. Let's look at another question. You need to explain how a double-blind trial is used to test a new drug. So try and remember what double-blind means and different things that you might have involved there. This time you've got 4 marks. Pause the video and give it a go. Okay, let's look through our mark scheme. So, because we're testing a new drug and we've got past the healthy people level, so now we're going to be looking at the clinical trials phase 2 and forwards, where we have small groups of volunteer patients. So we're looking for 2 groups of patients with a condition, and you're going to be using placebo on one group and giving the treatment to the other group. This means that you can compare the effect of your placebo against the actual drug that's there. And in place of placebo, you could also mention dummy pills, sugar pills, or fake pills. Because we know that placebo effect is something that can affect someone's outcomes, you need to mention the fact that there could be a placebo effect showing an improvement. You could also say that in a double-blind trial, that double-blind part comes from saying neither the testers or the scientists nor the patients know if the treatment contains the drug or not. This is going to remove bias, and it allows you to test to find out if the drug was effective. So from any of these points, see if you've got 4 of them and mark yourself out of 4. Let's have a look at one more, and this is summarizing the system of William Withering's time to our current system. You have a few gaps that you need to fill in, so I'd like you to pause this slide, have a read through, and then we'll go through the answers together. Okay, let's look through our answers. So to begin with William Withering, he has a potential useful substance that he's identified, but not a single active ingredient. We haven't eliminated all the other components in that plant, we just know that this plant extract contains something that's useful. And then he skips a bunch of tests that we would do, and he trials on a small group of patients with the disease straight away, as soon as he found it out. After he'd done that, then he used a large group of patients where the dosage was refined and method of extraction improved. And then straight to general use. Now let's look over the other side. This time, we know that there's something useful in the extract, but we need to identify the active ingredient first. And then, if it's possible, we're going to manufacture that active ingredient just by itself to make sure it's chemically pure. Then the next stage of our investigation would be to run preclinical trials on animals or cells to determine their safety or toxicity or their efficacy, or how effective they are. Next, we do the stage 1 of clinical trials, where we take a small group of volunteers and we're using it to check that the drug is behaving as predicted, so we're testing for side effects by the preclinical results. After this, you'd review your data, you'd look back through everything you've got and do some more checks. Then you start stage 2 of clinical trials, where small groups of patients with the disease are treated to see if the drug actually works. Following this, we can have the stage 3 of clinical trials, where we have a large patient group, where we're using a placebo, and patients are randomly assigned to a drug or a placebo group in something called a double-blind trial. And this allows us to statistically test if there is a significant result. After this, you can get approval for use. So let's bring both things together now. We want to compare and contrast William Withering's way with our current drug development timeline. I would like you to pick out some similarities, some differences. In what ways is the current system safer? In what ways is the current system more reliable? What we gain from testing on healthy volunteers first, and why you would need to assign patients to treatment and also have a double-blind trial. So pause for now and let's answer these few questions. Let's start with some similarities. So, in both pathways, you are starting with a potentially useful substance, and then we're going to small groups, and then scaling up to larger groups. And both William Withering and us today will try out different dosages to make sure that we find the most effective one. However, there's a lot of differences. For the beginning part, William Withering would just use the extract while we try to refine a single active ingredient. We use animal trials first, and then we review data an awful lot more, and then we also test on healthy people who have volunteered before patients. And comparing William Withering's hospital to the current clinical trial methods, when we hit that stage 3, you have a much larger sample size, you're using placebos, you've got double-blind trials, statistical testing, and we're doing all sorts of things to remove bias by randomly assigning people to groups, and then not just accepting, oh yes, we've made a medicine now, this can just stay out forever. No, you monitor your patients and you make sure that they are reporting any new side effects that might occur over the long term from taking this medication. So in what way is the current system safer? Now, by being able to refine a single active ingredient or a specific group of active ingredients, we can give a much more precise dose of it to our people. And by doing animal trials at the beginning, if there are serious side effects, we're not going to be giving them to this medicine to sick patients first. So by giving it to animals, we can see the side effects before they cause any harm to people. And to make the system more reliable, we're talking about repeats. So our repeats here come from having very large sample sizes to remove the effects of someone recovering just by chance. You're also doing double-blind trials to prevent the researcher from influencing the outcome, because you inadvertently might make decisions throughout the study or through reporting results where you are expecting to see a particular outcome, and so that might influence what actually is reported. And also through randomly assigning your patients to different conditions, you're removing bias that could be made in choosing people for different groups. And finally, statistical analysis will really help to show if there is a significant result overall that does make a difference, and also it will very clearly show if you have a drug that doesn't make a difference. So next, let's look at our last few. What do we gain from testing on healthy volunteers first? So the answer for this is that we get to see side effects, and it will really show us how long it takes for that person to break down and eliminate the drug from their bodies and just process it. And it's important to assign patients and treatments to double-blind trials, again because of the effect of bias. So your researcher can't ask loaded bias questions when they're trying to ascertain how somebody is feeling. They're not going to be showing through their body language indirectly that they do or don't expect an improvement, and this will stop patients from acting in a way that they feel they should based on the treatment given. So if somebody is asking the patient a question in a certain way, they might think that they have to give a particular answer to please the person asking them. So I'd like you to go back through and highlight the key points from those last few answers. So what keywords do you need to include? What new concepts, or what is the main idea behind this? And it's really important to draw it all together again through some past paper questions. So here we have a nice quick 2-marker. Medicinal drugs undergo clinical trials before they are licensed to be used. In January 2016, a clinical trial of a different drug involved 128 healthy volunteers aged 18 to 55. In this trial, different doses of the drug were given to 90 of these volunteers, and the others were given a placebo. 6 people who were given the drug became ill and had to be treated in hospital. So this question starts by asking you why healthy volunteers are given different dosages of the drug or the placebo. 2 marks, 2 points, give it a go. Okay, now you could have any of these points. We could say to check for side effects. You could also say the different dosages are used to determine what is a safe dose. Or you could mention the placebo and saying the placebo is present to make sure that any side effects described were due to the drug. Score yourself out of 2, and let's move on to the next question. The trial tested increasing the dosage of the drug. The 6 people who fell ill were the first to receive higher dosage over the course of several days. Which of the following is the correct ratio of those who took the drug to those who fell ill? You might need to go back and have a look at some more numbers. So, which of these ratios would you say? Okay, let's have a look through. So our correct answer is B, 15 to 1. So the explanations that are given in the mark scheme refer to the 2 numbers. We've got 90 volunteers in total, and 6 of those became ill. So the way that you could express this as a ratio before it's simplified is 90 to 6. So in order to simplify this to get 1 on this side, we're going to divide the whole thing by 6. So this will give us 1 over here, and 90 divided by 6 gives us 15. So according to our mark scheme, we should go for option B. Let's look at the next question. We have to compare and contrast drug testing protocol used by William Withering to the previously explained one. So there's 3 points that we can say comparing the modern case clinical trials and William Withering's Digitalis soup. So thinking back to that activity that we did earlier, comparing the 2, we could pick out that both trials use different dosages as a similarity, and then also to contrast it we need some differences. So William Withering did not use healthy volunteers, and this drug trial did. Also, William Withering did not use a placebo, whereas this trial did. There are your 3 points. So score yourself out of all of those marks in total and give yourself a summary. To bring everything together today, you need to be able to describe William Withering's discovery. We need to explain the main phases of his clinical testing and our clinical testing. We also need to compare the similarities and differences between his study and ours. So grab a post-it note or just jot something down on your word processors or paper if you've got a bit nearby. Compare those 2. If you have any questions, feel free to send me an email, and look forward to seeing you later.